Thank you, Andrea, for your kind introduction. And good morning, everyone. We have heard that cellular senescence was mentioned a few times in the previous talk. So my talk will focus on the effect of nutraceuticals on cellular senescence in animal models and humans. Cellular senescence is one of the hallmarks of aging and defined as a state of stable exit from cell cycle. It can be caused by multiple factors and is featured as growth arrest, DNA damage, lysosomal alteration, and the secretory phenotype, which is termed as CESP. Cellular senescence has been associated with many age-related diseases. More and more research focuses on xenotherapeutics, including senolytics and xenomorphics, to promote healthy aging. Nutraceuticals that derive from natural products showed benefits in prevention and treatment for age-related conditions by reducing senescence load and inflammation, so they have potential to be used as xenotherapeutics. With a focus on dietary ingredients, we did a systematic review to overview their association with cellular senescence in animal models and humans. We searched comprehensively in PubMed and Embase using the key terms cellular senescence, relevant markers, diet, food, nutrients, etc. But we didn't search for CESP because it has a wide range of molecules and each of them is quite often measured in non-senescent context. We included original research if they use dietary ingredients as interventions given through gastrointestinal tract and measured any of the cellular senescence markers. We excluded fasting, caloric restriction, as they are dietary behaviors which are not the focus of this review. And we also excluded FDA-regulated drugs in vitro studies, or if the study only had CESP measured, we also excluded. Here comes the result. If the intervention has a greener background, that means it has a higher evidence to reduce senescence. The top three ingredients, resveratrol, vitamin E, and the soy protein isolate, had xenotherapeutic potential. And if you see the middle panel here, the majority of interventions fall into the moderate quality of methodology, which means they were only investigated in 1 or 2 studies, although they have measured more than 2 senescence markers. Ginsenoside was investigated in both animal models and humans, showing a lower evidence to reduce senescence. Omega-3 fatty acids and the fish oil, which mainly contains EPA and DHA, showed no effect on senescence. And we can clearly see a huge gap of translation here, as the evidence in humans is quite limited. Some animal studies had a high risk of bias in incomplete outcome data and selectively outcome reporting, whereas the rest of studies had an unclear risk of bias, as none of the studies reported on allocation, concealment, random housing, and blinding. All 5 human studies had an unclear risk of bias in allocation concealment. On the other hand, what is the first diet or dietary ingredients you think of when it comes to inducing cellular senescence? We found that high-fat diet can induce senescence across all physiological systems. And excessive nutrients, ethanol can also induce senescence. Additionally, we have bioactives inducing senescence in tumors, but their impacts on normal cells from the same model remain unknown. One human study showed cellular senescence increased in the cells treated with serum from healthy participants after having red meat-based meals. And the other 2 human studies are RCTs, and one of them focused on high protein diet in muscle from healthy participants after exercise, and the other one is DHA-enriched fish oil in PBMC from diabetic patients, and they showed a trend of inducing senescence. Xenotherapeutic effects can be varied in different models, biomaterials, and the dosage used, and studies that were insufficiently reported or poorly designed, conducted, or analyzed really pose a challenge to the translational reliability. Thus, to facilitate translation, high quality and better designed studies that following relevant guidelines are needed. In conclusion, while highlighting the translational challenge, we identified promising xenotherapeutics and the dietary interventions that are shown to induce senescence. And these findings suggest the potential application in clinical nutrition plans as the strategies that aiming to mitigating aging processes. So I would like to thank the team, Anna Christina as the second reviewer, and Alina Karen as the third reviewer, and also many thanks to my supervisor, Professor Andrea Meyer, for her guidance and the support. Thank you very much for your attention.